Hi, and welcome to the Davos in the Desert podcast series. These podcasts feature thought leaders in business and public policy and are meant to be informative and thought-provoking. In this podcast, we learn how stem cells are harvested and modified into regenerative therapies. Understand the difference between stem cells arising from umbilical cords, placentas, blood, and a variety of organs. Listen to which stem cell therapies have received FDA approval and which ones are close to doing so. Gain insight into a variety of government programs designed to stimulate the development of stem cell therapies. And know where the most restrictive bottlenecks for producing stem cells exists. Without further ado, here is David Waintick, Davos in the Desert CEO and our podcast host.
So, Dr. Jane Andrews, thank you very much for joining us today.
Thank you for having me. I appreciate the opportunity.
So, we're talking about stem cells. There's a lot of interest in stem cells, but many of us don't know why we're so interested in stem cells. So, maybe you could just take a couple minutes and give us a nice overview of what stem cells are.
Stem cells are cells that regenerate themselves. And they regenerate the organs that they're supposed to be regenerating inside the body. Many years ago, when we discovered what stem cells were, I was a researcher at the University of Wisconsin-Madison, and I was working in mammalian or animal reproduction. And we were working with pre-implantation embryos. Just a ball of cells, a ball of 16 to 32 cells before they implanted in the mother. We were looking at them in the petri dish, trying to figure out how they operated. And it became clear to researchers, not myself in particular, and I wish I had figured it out, but you could take one of those cells, just one of those cells from that pre-implantation embryo, and you could put it in a new mommy, and it would grow into a whole new baby. That is what we call a totipotent stem cell. That is a cell that has the ability to create a whole new individual from itself. So those cells in that 16 to 32 cell embryo were all totipotent. They hadn't differentiated enough yet so that they had specific functions. And that was phenomenal to learn that. And then in the late 90s, there was a lot more interest in this in human. But the government really wasn't, from a moral, ethical standpoint, didn't want to put any federal funds behind that kind of thing. So there was kind of a moratorium on cell therapy research for quite a few years, until we started to realize that adults, you and me, we had stem cells in our adult bodies. So we have stem cells for our heart. We have stem cells for our nerves. We have stem cells for muscle, for bone, for blood. They're adult stem cells, and that's what we study mainly today, which has opened up this whole new field we call regenerative medicine. And regenerative medicine, in other parts of the world, it's called advanced therapies. It's called cell gene therapy. So there's a number of different names, but overall, it's regenerative medicine. And it involves the study of a number of cells that are regenerative.
Okay. So when stem cells replicate or regenerate so much, to me, it may sound like a little bit like what cancerous cells do. Is maybe you can differentiate the processes?
Yes, there are cancer stem cells that actually kind of go wild. They lose their regulatory mechanisms and they replicate without control. And that's one of the things that makes them cancerous. And they are bad cells and their metabolisms are very aberrant. So that is one type of stem cell, but that is not the norm. The norm would be a stem cell for your own nerves or your own, for example, bone marrow. When people are very sick with leukemia or some sort of disease where their blood is in very bad shape and has cancer, they'll take that person's blood out and they'll do a bone marrow transplant because it contains healthy blood stem cells. And that person then can create healthy blood stem cells. That's one of the main sources of adult stem cells in your body.
Okay, so if an individual has bone cancer or heart problems, you could extract some stem cells from the heart or from the bones. And what happens after that? How are they treated before they're reinserted back into the patient?
Well, that varies depending on the cell type. So what I'd like to do is focus specifically on what regenerative medicine is, or I'll give you a definition before we go into how they're biomanufactured, because biomanufacturing is a key point in the development of this whole industry. Regenerative medicine is what we'll call the next phase of medicine. It's leading edge. And as any new medicine that's leading edge, there's always sort of what we call a green field, where there's a lot of, everybody's doing something a little different. And so we have to get everybody on the same page doing the same thing, so the same product is created for the individual. So really one of the roadblocks in this new industry is biomanufacturing. And this was the case with small cell biologics as well, 25, 30 years ago, before we learned how to create vaccines in large vats very, very efficiently. Every new science that comes on board, there's always this startup issue that kind of occurs. So let's just go to where we can define what regenerative medicine is. If you don't mind, I will bring up a slide. Can you see the slide?
Yes.
Okay, so this is the definition. We can substitute in regenerative medicine for advanced therapies or vice versa, but I'll just stay with advanced therapies here. The advanced therapies includes all tissue and cell therapy, cell gene therapy, immunotherapy, and cellular components such as exosomes. Advanced therapy is a branch of translational research in which tissue engineering and cellular and molecular biology are used to replace, engineer, or regenerate human cells, cellular components, tissues, or organs to restore or establish normal function. That's a huge definition. It's in fact, it's just so huge. It involves a number of different types of therapies. It's gene therapy, cell gene therapy, tissue therapy, combination therapies, exosomes, which you can get into in a minute if you'd like, or there's now even synthetic biology that's being investigated. But before we go any further, what I would like you to know is that as of April 9th, 2024, the FDA has made a public statement indicating that currently the only stem cell products that are FDA approved for use in the United States consist of blood-forming stem cells, also known as hematopoietic progenitor cells that are derived from umbilical cord blood. Okay, so stem cells in themselves are still under research. There are blood-based cells there that are used as products and have been approved. There's 37 approved products that are immunologically based products. They are cell and cell gene products that have been approved for use. And we'll go over some of those as well. We just don't want to get the general population thinking that there's a whole number of cells out there that are approved, stem cells that are approved by the FDA for use in clinics around the United States, because that's not the case.
If I can just ask one more question, I think you said that a lot of the research now is coming from the stem cells from adults. Is there an age, generally speaking, at which the stem cells are no longer so useful?
As we get older, our stem cells age. And so the older or the sicker the individual, one of the two, the less functional those cells are.
So can that individual maybe ask a family member to harvest cells from that individual and then?
That is the case in some cases, but what we have is, as you know, we have we all talk about rejection issues, right? If you have an organ transplant, you're worried that that patient's going to reject that organ. You can also have the same issue with rejection of stem cells from another individual. So there are two categories. There are allogeneic and there's autologous. Auto means self. So you take stem cells from yourself and you put them back in yourself. Okay, so you can the doctor can take cells from you, treat them, and put them back in you. And you don't really have a rejection issue. With allogeneic, you're taking cells from somebody else, treating them, and putting them in another person. And there is sometimes a rejection issue. The areas where there's least likely to be rejection is if you did a bone marrow transplant from a family member, and that's been done for years and years and years. Or there's perinatal cord blood. Cord blood is what we call immunoprotected because it comes from mommy and baby. And that cord blood doesn't want to be rejected by the mom, and it doesn't want to be rejected by the baby. So we call it immunoprotected. So of those 37 products that are approved by the FDA, over 8 of them are from cord blood.
I'll let you get to your presentation in just a minute, but just a thought that came to me. For manufacturing of food, manufacturing of synthetic meat and so forth, is some of the science similar to stem cells, stem cell growth?
I am not familiar with manufacturing of synthetic meat, so okay. I can't address that. I'm sorry.
Okay, all right. So
I would not think so, but I don't know what the process is. So there are these different cell types that are considered advanced therapies. And what makes this so difficult is it's a convergence of technologies. It's like nothing else we've ever done in science. Usually if you're studying cardiovascular disease, you have cardiologists, you have physiologists that are heart specialists. If you're a neurologist, you have people who are focused on one discipline. The advanced therapies, regenerative medicine, takes people from many disciplines and brings all that information together to make a medicine. And getting all of this information together is where you get the bottleneck. And so if you look at the lower right-hand picture, you see a bottle and there's a bottleneck. And that is because manufacturing is the bottleneck for biomanufacturing of regenerative medicine products. So when we go on here further, we can see that you asked me about, is there a time when your stem cells aren't functioning anymore? So that's a really good question because it gets into the idea of populations and why we need or want regenerative medicine. The global population is aging. Today, there are many treatments for chronic or life-threatening diseases that are palliative, which means they don't cure the disease, they only treat the symptoms. That's especially the case with cancer. We have what I call a silver tsunami of an aging population. Globally, there are already more adults over 60 than there are children under 5. And by 2030, there will be more people over 60 than under 10. In the US, by 2030, 19% of the population will be over 65 years of age. And in Japan, already 25% of the population is over 65. We also have a thing called socialized medicine, where governments pay for healthcare. And the older people get, the sicker they get, and the more expensive it is for governments to keep them healthy. There are countries that are going bankrupt because of the cost of healthcare. So one of the goals of regenerative medicine, and one of the reasons many countries are behind it, is so that you can keep older individuals healthier longer. It's one of the drivers of regenerative medicine. So if we think about regenerative medicine as an ecosystem, we think about all the things that are brought together. Like you saw that roadmap where it was a convergence of technologies. This is kind of a circular idea of that ecosystem. So if you think about your starting product are cells, right? So you take those cells from yourself, that would be autologous, or from if you were sick, or someone else, that would be allogeneic. But this is the starting material is cells. So they have to be collected through a process very similar to how blood is collected for blood collection and transfusion, right? A process called apheresis. Those cells have to be looked at to see how healthy they are, how sick they are. You have to put them through a quality management system. And I mean, you look at the quality, you have to control everything. People are in suits. This is all very strict, high quality airflow, no diseases, no cross contamination. It's very sterile environments. Sometimes they're genetically modified. So, for example, if you have a leukemia of one type, you can find out what type that is, get the genes that will treat or kill that type of cancer, and insert those genes into the cells. So you're genetically modifying the cells to treat the cancer. We call that cell gene. And many of those are the ones that have been approved by the FDA because they have such high efficacy. What's happening here is we're using those genetically modified cells to cure cancers that were never cured before, ever.
So to actually extract cells, let's say from the heart, is that a surgical procedure? How do you actually get into the heart?
Usually you do not treat the heart with heart cells. Most of the ones that have been approved by the FDA have been blood-borne cancers. Okay, so if heart disease has not been one that has been, there are some heart clinical trials in progress, but they are not the ones that have been approved by the FDA as of yet. That's a very good question. There's a lot of research that's being done on that. There are cardiomyocytes that are a stem cell for the heart is called a cardiomyocyte. They're being grown up in culture, and they're being looked at and how they can be modified to treat heart disease.
How would they be made specific for the patient? If the culture is grown in the laboratory and I need it, how would it be modified to help me?
It would be dependent on your type of disease. So whether that is not really something we will be addressing here, because each of that is an individual case. So what I'd like to really emphasize with this diagram is that a lot of blood-borne cancers, like leukemia and even lymphomas, have been cured by genetically modifying a person's own T cells, immunological cells. So you collect the cells from the person. They may be genetically modified. In some cases, they're not genetically modified. They're processed. The good ones are picked out and kept. And then more of them are grown in culture. So you have to get enough of them to put back into the patient. And once there's enough of them, we call that expansion. And they're then stored and then they're transported back to the patient. Oftentimes, this whole process is done off-site, away from the hospital, in a biomanufacturing facility. And there are many, many hundreds of steps here. And the more manual this process is, the less likely it will be to succeed. Because the more human touch points you have, the more probability you have for error. So what we're trying to do now is make much of this automated as possible, because you have to look at the cells and have maybe a sample taken out automatically and looked at to see what quality there is there. And then you can go on and change the system based on quality before it goes back into the patient. So why do we do this kind of thing? This is the first child that was ever treated with Kymriah, which was a cell gene product like we were just talking about. And here she was, very, very sick little girl. And Carl June met her, and she was going to die. She was already given her last rites. She was 5 years old, diagnosed with acute lymphoblastic leukemia, and she'd been treated for 2 years unsuccessfully. And Carl said, well, let's try this brand new treatment, a cell gene treatment called Kymriah. It was a 1-dose treatment. There she is last year in May, completely cured, completely cancer-free. First time ever that this has ever happened. So therefore, and this has happened time and time again, this Kymriah was one of the first cell gene products to ever be approved by the FDA. And I've already gone over what autologous and allogeneic is, and we'll just go over some of these. Now, I told you there were a lot of different cell types. Some are auto, which means you can take them from yourself and put back in yourself. Others are taken from other individuals and put back into other individuals. So these are the different cell types that can be used. These are different T cells, which are from your immune system. These are stem cells from different parts of your body. And these are mesenchymal stem cells, which are also in your own adult body, from adipose tissues, perinatal tissues, bone marrow, peripheral blood as well. So you can see there's a lot more of them that work when you put them back in yourself than the ones that go into another individual, because we have that rejection issue.
So are all of these FDA approved?
No, all of these are not FDA approved. The only ones that are FDA approved are the ones that are the cell gene products. Stem cell products are not approved by the FDA. Now, one of the reasons for that, you and I spoke about this earlier, is that physicians would, and around the world, physicians can take cells from a patient in their office. They can modify those cells and put them back in that patient. And the FDA has a rule now that you cannot do anything more than minimally manipulate those cells before you put them back in the patient. If you do anything more than that, it's called biomanufacturing. And if that's the case, it has to go through full FDA drug approval. So stem cell products are not approved by the FDA in the United States. The only ones that are approved are the ones that are from your blood for hematopoietic bone marrow transplants, which have been approved for years. And then ones from the placenta to put it back in children.
So are the big pharmaceutical companies putting a lot of research into stem cells, or is most of the research done
Absolutely.
at universities or small companies? So the big companies are involved heavily.
Big companies as well as universities are putting a lot of money into this. Allogeneic alone. And allogeneic, which is basically, this is a $5 billion market. And this is for hematopoietic stem cells, MSCs, and induced pluripotent stem cells all together, which is all of these types. And this is basically the academic portion alone. It's not even including the large pharmaceutical companies. And it's growing at a fairly rapid rate of a 7.7% CAGR. The pharmaceutical companies are putting a huge amount of money into more of the cell, gene, and advanced therapies as well. And I don't know if I've got those values here. But it'll suffice to say that, yes, here we go. This is the market size in the US alone for autologous, meaning from you back to you. Globally, the market in 2023 was 7.3 billion. It's calculated to grow at a rate of 19.4% to reach 25.1 billion by 2030. And in the US, that number is 10.6 billion by 2030.
Are these research dollars directed towards these applications, or are these revenues from products?
This is money put into the market. This is not revenue generated by these. This is money invested. This would be VC funds, it would be investment dollars, pharmaceutical funds, money put into the industry. So very good question. So you can see there is a huge promise for this because we have seen such high efficacy. In fact, the FDA has stated the problem with cell therapy is not efficacy. It isn't its ability to cure. It's the biomanufacturing process that's the bottleneck. So a lot of effort needs to go into making this more streamlined so that we have a better biomanufacturing process.
So is the bottleneck within that there has to be almost 100% customization for any solution, or does it produce a pill?
No, there does not have to be 100% customization. That's what we're trying to get away from. There has to be more, less customization and more automation.
So what can go wrong during the automation that the FDA is so concerned about?
The FDA is not concerned about the automation. The FDA is concerned about the fact that there's not enough automation. And what goes wrong is that the product isn't of high enough quality to have high enough efficacy and repeatability.
Well, that would seem to be a very good area to invest in if there's.
Yeah, absolutely.
So, of all those big numbers you showed us for research and development, any ideas how much is going to like fundamental research versus production and biomanufacturing?
Yes, those numbers are increasing as you and I speak. There's more money going into the biomanufacturing process.
And are these like dedicated companies that are popping up to do the biomanufacturing, or are the big pharmaceutical companies putting those resources in place?
There's the government as well as large pharmaceutical companies that are putting money into that. So there's the equipment that's involved as well. So there's companies like Terumo, Sativa, PAL, Thermo Fisher. There's Samsung's very interested in getting involved in that. Catalent, a lot of companies. That's just a few. It's almost every pharmaceutical company you can think of is putting more funding into making this biomanufacturing process more efficient.
So A lot of parents are preserving stem cells when babies are born with umbilical cords or placentas. How long do those stem cells last until that baby is 80 years old? And when the baby is 80 years old, and they have heart problems or bone cancer or something, can those same cells be used 80 years later to repair the damage?
Theoretically, they should be because they're stored in liquid nitrogen. So theoretically, yes. The concern we have is that there won't be enough of those cells to treat a large patient. The umbilical cord only has so many cells in it, and it in the past has only been able to treat a patient the size of about 57 pounds, because it hasn't really been easy to make more of them, expand them. And technologies are now getting better. There have been 4 different types of treatments in clinical trials to expand those cord blood cells so that you can treat a larger person.
Okay, so what's happening there is the cells are replicating
Yeah,
outside of the body in the bank.
well, you would thaw them out,
But
and then you would make more of them,
okay,
and then put them in the adult.
so does if would it be a good strategy for a parent to have some of those cells replicated shortly after they're put into the bank, or before they're put into the bank, try to replicate more of them at that point, or wait till the baby's 80 years old and then try to thaw them out and then replicate them at that point?
It depends on the technology at hand. From a physiological perspective, it may be more advantageous to wait because technology will be so much more advanced 80 years from now that it's much more likely that they'll have the technology needed to make the product that's needed for the individual.
Seems like a stem cell bank might be able to quadruple its revenues if they would, offer a program to replicate the cells as they're collected, just so they could say, well, when the patient's 200 pounds, we'll have enough. 4 times as many are being donated.
Again, that depends on the state of the technology at the time and what you're trying to do.
So, in a very simple understanding, an adult has a cancer.
I'm sorry?
Let's just assume that an adult has a disease like leukemia or heart disease. So, a lot of the solutions are blood-based. So you extract the blood and put it in a culture.
Yes.
And try to pick out the good cells within that blood.
Yes.
And maybe manipulate some of it genetically.
Yes.
But now the issue at the biomanufacturing is
that's part of biomanufacturing.
Okay. So
that's what we call biomanufacturing.
So is the bottleneck within that, is it skilled people, or is it equipment or capital that's the real constraint?
It's the cells have to be treated just a certain way. So they have to be cultured a certain way, they have to be selected a certain way, they have to grow. Cells, it's hard to grow cells in culture. I was a physiologist for many years at the University of Wisconsin-Madison and cultured many, many cells. Cells are it's an art. It's not like car manufacturing. It's completely different. So you have to have the right culture conditions. You have to have the right gas mixtures. You have to have the right nutrients. You have to, and then they'll only multiply so many times before they stop multiplying. Cells are very finicky. And so creating the appropriate conditions. Now, if a patient's, for example, if a patient's really sick and they've been treated with chemotherapy 3 or 4 different times, and that's your starting cells, those cells aren't very happy when you start with them. And then to get them to multiply enough to get enough cells to treat them on the back end downstream is very difficult. So a lot of this is technology based. It's also it's technically based and trained personnel based. You are very correct there. There are not enough trained people in the industry yet. And as I said, this is a convergence of technology. So you're putting these cells in we call a bioreactor. And the bioreactor technology will hopefully keep them alive longer and help them multiply. There's also techniques that Terumo is creating to collect the cells more gently so they're not damaged when they're collected. It's a very gentle centrifugation where you get rid of all the extraneous cells and fluids and things. And they've been very successful in their collection process. And then it's the freezing process. You know, freezing is notorious for there's been no real new freezing technology in the past 30 years. So once you spend all this time, it can cost you $500,000 to treat one patient. Because you're creating one product per patient. And you need a lot of technology and a lot of expertise. And therefore, you want to make sure everything is done absolutely properly. The bottleneck is all the steps that are involved.
Yeah, so the FDA probably feels it doesn't have the resources to inspect all these different efforts. Each one is like a separate research project. It seems like each exercise in manipulating the genes seems to be
the
its own.
FDA is very happy. In fact, it is so happy with the efficacy that we are now seeing. It predicts, Peter Marks has predicted that we will see 20 new products approved per year. And we are starting to see that already this year. It looks like we're gonna hit that target and maybe go beyond. They have in place the 21st Century Cures Act, which is a method for fast tracking some of these things. They also have another program called RMAT, which is helping to fast track some of these technologies through the pipeline. They have created a super office in 2023, and it has 6 sub offices. It has new leadership. It has new reviewers. Its motto is better communication. And it believes that the more touch points you have with the FDA, the better it is. And they've also created a new program called ARPA-H that focuses on life sciences, but also a number of regenerative medicine products. Okay. Some of the focus areas that the FDA will be focusing on is rare diseases and genomics. Personalized medicines, which is exactly what you and I were talking about just now, and accelerated approval. According to Nicole Verdum, who is the director of the CBER and the therapeutic products and the super office, it's better to have many touch points with the FDA as possible. So they understand that this is the new wave of medicine. There are many platforms, David, just as you had said, but there are ways in which you can get some platforms that are basically standardized, and then variations upon that platform.
Okay. Are stem cells also included in, I think it was like end of life program that under the Trump administration, I think the policy was that if
the right to try. Right to try. Is
yes. That covered here? Okay.
Yes.
Okay. Well, maybe looking at things that are not life-threatening, diseases that are not life-threatening. So you hear about people that have treatments of their knees or joints with stem cells. Is that type of thing FDA approved, or how safe?
Not yet, not yet. It is in other countries. It's approved by other countries, by their governments. There is some progress. There's one here that I saw with osteocytes, chondrocytes in a scaffold that had been approved just recently for knees, which is really exciting. And I think to be very honest with you, I can see a lot of promise in all of these things. Mesenchymal stem cells are really not stem cells, they're really stromal cells, but they really are helpful with inflammation. And they modulate the immune system. So there's a lot of promise for MSCs. The problem we have with them is we don't have, everybody uses a different platform to a certain extent, and we don't have enough continuity. That's what we need to do with biomanufacturing. We need to get better biomanufacturing so that we're sure the end product is going to be the same every single time. And that's why we're seeing sort of a lag in some of these other products going through the pipeline.
So for some of the things like knee procedures, can the stem cells come from other people for those procedures?
There's great promise for perinatal, like I said, because they come from mommy and the baby, and they're immunoprotected. So if you can collect placenta and get MSCs from the placenta, there's less rejection issues. Or if you can scientifically modify an adult MSC or chondrocyte, if the science advances enough so that you can immunoprotect those cells, then there's more likelihood of success long term.
So maybe, and winding up, maybe the last two questions, if somebody was going to bank their stem cells, what questions should they ask to determine the professionalism at that stem cell bank? It seems like it's the kind of thing that a scam artist could get involved with, because you don't have to produce anything for a long time. Many people are never going to call for their stem cells. I believe the contracts say there's always some risk of slippage or breakage, or there is some risk of problems, so they cover themselves with that. But if one was going to donate their cells to a stem cell bank, what questions should they ask? And similar for less life-threatening procedures, like knee treatments. If somebody's going to get a stem cell knee treatment, what questions should they ask the medical professional that's going to deliver?
Well, stem cell knee treatments are not approved in the United States, so I can't address that. I can't answer that question. The person would have to go overseas to have that done, unless they went with a professional that was kind of flying under the radar, which I really can't address that issue. With regards to banking of cells, you would want them to have FDA inspection. There is an FDA banking program. You would want them to be aligned with that. There's the AABB, which is an organization for banking of cells. You would want to make sure they are in line with the AAB procedures and protocols, and that they are well known for their banking standards.
Maybe in conclusion, why don't you just tell us a little bit about what you do when I'm not interviewing you at Stem Cell Bridge Strategies?
So I help companies evaluate their technologies and bridge the gap between science and business. So oftentimes you have academic companies that want to commercialize a product, but they have no business acumen, and they need to translate that science into a clinical trial, or get through clinical trials, or get them on the market. So I often help companies with my team evaluate their products and what they're doing, and help them build a roadmap for commercialization of that product.
So do everything from advising them on which CROs to use to market strategies. Do you help with getting insurance coverage for products?
I do not work on the payer access. If we do have someone who is working in that area, we bring people on board, on staff, to help with the payer access. We basically help them with the process of getting enough product so that they can get through clinical trials, through the commercialization process, and that they have a viable product coming through. We do not help them get that on the market per se. So some of our clients have been big pharma all the way down to small startups. So if we've worked with big pharma that already have a product that say it's not selling well on the market, what do I do differently? So we'll go out, we'll ask people that are using their product and find out what they like or don't like about it, and go back and help the company modify that product to make it more user-friendly or hit the market better. We help identify competitors. We help them position their product in the market. We help them identify ways in which they can get through clinical trials more readily and at a lower cost than they were thinking of doing. We help them build the team they need, help them look at return on investments, help them look at their biomanufacturing processes, bring partners in to help them get through their hurdles, help them build an advisory team. Many of these companies can't do it on their own. Everybody needs a little help.
Sure, sure. Well, Dr. Jane Andrews, thank you very much for spending this time with us and educating us on stem cells. This is a difficult topic. We hear a lot about stem cells, but it gets very complicated very fast. So glad to have an expert such as yourself speak to us.
Thank you.